Hi everyone, welcome to Journal Club. Make sure you fill out the attendance and we're gonna get started. So welcome Hannah.
So today I'll be presenting on the neural code of anhedonia, which is a stress response breakdown. So to provide some background information first, this study investigates how stress alters neural population activity, which is the activity of many neurons grouped together and working together that underlies anhedonia. And previous studies have focused on single neurons or isolated circuits, but depression likely involves population-level neural dynamics across regions. So anhedonia is the reduced ability to feel pleasure or desire rewards, which is a core symptom in approximately 70% of all patients with depressive disorders. And chronic or severe stress is a major risk factor for depression, but the neural mechanisms that determine resilience versus susceptibility are not well understood yet, so this study investigates that. And while there's no exact number for suicides caused solely by anhedonia, research consistently shows a strong link, with anhedonia being a significant independent risk factor for suicidal thoughts and attempts, meaning many people who die that way experience it. And it's truly a life or death matter, so the research being done in the study is very important and could lead to very meaningful discoveries. Researchers recorded neural activity from two key regions in reward and stress processing of the brain. First is the ventral hippocampus, also known as the VCA1, which is involved in contextual memory and stress processing, as well as the basolateral amygdala, also known as the VLA, which is involved in emotional and reward-related information. So this study uses a chronic social defeat stress, also known as the CSDS mouse model, to distinguish stress-susceptible and stress-resilient individuals based on behavior like sucrose reward preference and social interaction. And researchers aim to quantify anhedonia and verify that neural changes had functional behavioral effects. So first is the social interaction experiment. To test that, they create a measure for anhedonia. Over the course of 10 days, each experimental mouse was introduced into the home cage of an aggressive mouse for 10 minutes of direct interaction. So they experienced physical defeat, or known as bullying, and then moved away for 24 hours, allowing sensory exposure. And this repeated social defeat experience induces social stress and similar depression-like states in a proportion of animals. Researchers tested a mouse for social avoidance or interaction behavior by placing one in an arena with an empty enclosure and recorded baseline exploration for 2.5 minutes. And they then repeated this, but replaced the empty enclosure with one with a new aggressive mouse, but not the same one as the previous bullying. And then they observed the results. Susceptible anhedonic mice showed reduced time in this interaction zone with aggressor mice, avoiding social stimulus, while resilient mice did not show social avoidance. And they also did an experiment to test reward sensitivity and create a measure for anhedonia. On each trial, mice are presented with two spouts, delivering either a sucrose solution, which is the reward, or water, which is neutral, and a cue signal trial start, and then mice choose by licking one spout. And the task cleanly separates reward value to the sucrose or water, while allowing precise measurement of licking behavior and neural activity. And after chronic stress, mice are classified as control, resilient, or susceptible. Resilient mice showed a strong preference for sucrose, similar to the controls, but susceptible mice showed reduced sucrose preference, decreased reward sensitivity, which indicates anhedonia. And so this is figure 1 from this paper. It shows a heat fix preference task combined with neuropixel recordings from the BLA and BCA1. Panel H, which is up there, compares lick rates for a water and sucrose. The water serves as a control, and you can see lick rates remain similar for all three groups before and after reward delivery. However, susceptible mice showed lower sucrose lick rates after reward compared to resilient mice, which indicates reduced motivation or pleasure in actually consuming the reward. And then panel I over here depicts the discrimination index of lick rate to differentiate high value, which is the sucrose, versus the low value, which is the water rewards. And you can see weaker discrimination between sucrose and water. The blue line here represents susceptible mice is significantly below the gray line, which is representing the control, and the yellow one, that's representing resilient mice. So susceptible mice discriminate less between high value and low value rewards, which is the hallmark of anhedonia, where the subject fails to value or preferentially respond to reward. And overall, figure 1 validates that stress produces distinct behavioral indicators of anhedonia, reduced reward seeking, and reduced valuation of rewarding stimuli, and form the behavioral foundation for the neural analyses that follow in this paper. And if you can see, there's some like stars over there, and those represent that the findings are statistically significant from the control and are unlikely to be due to chance. And like the more stars. This is figure 2 of this paper, and it depicted neural activity in both the BLA and the VCA1 during a sucrose preference task designed to assess reward-seeking behavior. The x-axis down here shows the 3 mice groups, which are control, susceptible, and resilient. And researchers found that resilient mice had the highest proportion of reward choice selective neurons in the BLA during both the time before reward delivery, which is like the pre, and then after, which is the post. Susceptible mice showed a lower fraction of these selective neurons in comparison, which is like visible in the graph. And this suggests that resilient animals maintain strong neural discrimination between the high-value sucrose and low-value water rewards in the BLA. And this difference is neuronal selectively underlies the behavioral differences in reward seeking and therefore anhedonia. So the results from this figure support previous data and the researchers' original hypothesis that stress resilience preserves reward coding. So these are Markov models, and Markov models work by predicting the future behavior of a system based only on its current state, and they use the probability of transitioning from one state to another. Like an example would be predicting tomorrow's weather based only on today's. And in the experiment, researchers found that the current and previous reward choices are not independent of each other. Markov models show the probability that mice will choose sucrose or water depending on the choice from the previous trial. In susceptible mice, they were more likely to switch from sucrose to water across the trials as compared to the resilient or control mice. And they additionally showed a greater consecutive preference for water, not choosing the reward. And then here you can see a hidden Markov model, which has some unobservable hidden states, but the model still creates outcomes that are observable. And from those hidden Markov models, in our experiment, it was found that the BLA of selective mice had a higher fraction of intention selective states in the seconds before the reward. So figure 3I presents us with the decoding of the switch versus stay decision in those susceptible mice. It shows how well neural activity can be used to decode reward choice, sucrose versus water, from the population activity, and how this differs across groups. On the x-axis, as usual, you can see the 3 mice groups, which are control, susceptible, and resilient. And then this time, the y-axis shows decoding accuracy with values between 0 and 1. A value of 0.5 represents accuracy by pure chance for either switch or stay, which we can see in the control mice. The researchers trained a classifier to distinguish where a mouse will switch reward choice or stay with the same reward based on the neural population activity before the reward is actually delivered. Resilient decoding is slightly above chance, just a bit under 0.6 if you look. And it's marked significant with like a hashtag, but still weaker compared to the susceptible mice, which is the blue. Decoding in susceptible mice is significantly above chance, the highest of all 3 groups, and the BLA population activity contains predictive information about the intention to switch or stay, which may underlie anhedonic behavior. And essentially, what this figure shows is that the Markov model described earlier produced accurate predictions of susceptible mice behavior, and it shows a strong connection between their behavior and the neurons in BLA. So the authors had already shown a correlation. Mice that were more resilient showed stronger functional communication between the VCA1 and BLA, but however, correlation alone cannot tell you whether that circuit activity actually causes resilience. So to test causality, the researchers intentionally manipulated the VCA1 to BLA pathway and measured behavioral outcome and neural population decoding. They showed that altering the circuit directly changes both of these, proving causation. So VCA1 to BLA communication is not simply associated with resilience, but it actually plays a mechanistic role in producing it. CNO, which is a clozapine and oxide, was injected into susceptible mice because it increases the excitability of neurons fired from the VCA1 to the BLA. which essentially means that they made it easier for them to be fired. The x-axis shows the experimental conditions after circuit manipulation. So salien's the control experiment in blue, and then CNO is the one in red. And each line here represents one mouse's change in sucrose preference from saline to CNO. And as you can see, there's more like upward sloped lines, which indicates an increase in sucrose preference, which is how the researchers are choosing to measure anhedonia in mice, shown as a percentage on the y-axis, compared with previous performance with saline in these mice that had been stress susceptible. And the circuit intervention is successful in reversing this anhedonia symptom, not just brain activity. There were quite a few significant findings and conclusions in this experiment. So researchers observed distinct neural activity in the resilient mice versus susceptible mice. In resilient mice, their brain activity mainly reflected how rewarding something was, which helped them choose good outcomes. Versus in susceptible mice, their brain activity was stuck on past decisions, whether to switch or to stay, which looks like rumination and is defined as stress causing the brain to over-encode internal decisions, which is similar to repetitive negative thinking in depression. But this is not the same as what occurs in human rumination and interferes with normal reward processing. And by changing the signals sent from the VCA1 to the BLA, researchers were able to bring back brain activity to a healthy pattern and restore reward-seeking behavior in stress-susceptible mice. And these findings have significant implications moving beyond single neurons and showing that population-level neurodynamics encode vulnerability versus resilience to stress, which is more realistic for psychiatric conditions. Again, they also proved causality, not just correlation, as manipulating the VCA1 to the BLA input actively reverses both abnormal neural activity and anhedonic behavior. Resting state activity carries stress signatures, suggests potential for biomarkers that do not rely on behavioral tasks, which is relevant for clinical translation if they want to further in this experiment. And then researchers also provided a neural framework for rumination, which is a core symptom in depression, grounding it in measurable circuit dynamics. There's also quite a few limitations of this experiment. For example, chronic social diffuse stress in mice captures certain aspects of depression, but human depression is much more heterogeneous, so findings may not generalize fully. Anhedonia is measured through reward-seeking behavior in this experiment, which is what the researchers here chose, but that may not capture emotional or subjective components seen in humans. This study also centers on the VCA1 and BLA, but there's also other regions of the brain, such as the prefrontal cortex and nucleus accumbens, that also likely interact with this circuit. So while resting state signals are promising, it's unclear whether similar neural patterns can be reliably detected or manipulated in humans using non-invasive methods yet. Future directions. A key next step is determining whether similar population-level neurodynamics can also be identified in humans using tools like fMRI or EEG. Mouse behaviors and neural signals are only approximations of human depression and cognition. So future studies could examine how other regions interact with the VCA1 to BLA circuit to shape how stress responses. And this study primarily uses one stress model, the CSDS. So testing different stress types and including sex differences could also improve relevance and generalizability. But overall, this paper provides new insights into the mechanisms of anhedonia, which again is a core symptom in depression and other mental health conditions, which makes it highly relevant to neuroscience and clinical research audiences. I'd like to thank all the journal club members who helped me with this, as well as Mr. Maxwell, and all the teachers providing extra credit today. Questions? Joe?
So why did the researchers choose to study resilience instead of just stress?
Yeah. I think the researchers chose to study resilience because not all individuals develop strict depression-like behaviors from stress. So studying resilience can help identify protective neural dynamics and mechanisms rather than only like pathological ones. Michelle?
During the mice social interaction test, what did the researchers actually measure?
I think it was just measuring like actual avoidance as a social stimulants, not like playing or bonding with like other ones. Ivy.
It goes back to the last graph.
This one. Is it just like more okay. Okay. So looking at these individual lines connecting 2 points for each mouse, if you look at like the left endpoint of each line, that's measuring the sucrose preference being measured for when the mouse receives a saline injection, which is measuring like baseline behavior. And then the right endpoint of each line is the sucrose preference measured when the same mouse received CNO. So yeah, like an upward slope line shows that the mouse increased sucrose preference after VCA1 to BLA activation versus like a downward shaped line or flat. That means little or no increase, but that's rare in this figure because overall there was an increased trend. And I mean, I thought this was kind of interesting when I was researching this paper. This like specific type of plot has a name. It's like called like a spaghetti plot, and it's especially useful here because it shows that the effect isn't driven by a few outliers. Like it shows it.
Wait, Lucas has a question.
Was there like an ethics code that the researchers had to follow when carrying out these experiments?
Like bullying the mice.
Yeah.
Yeah, I don't. They didn't really mention it, but I feel like there definitely should be because like, repeatedly bullying mice probably is ethical. Yeah. What you said?
The drug that they used here, clonidine, is that used commonly in humans as well?
It's a great question.
And then
look into that.
my secondary question was, is this the only drug that they use to test the
Yeah.
rescue of the behavior?
Mhm.
Okay.
So, like, I guess for future studies, if they want to like keep exploring it, they can test more.
I know there are a lot of different drugs that are used in clinically for humans, right? So I'd be interested to know if this is unique to this particular drug or others also.
I'm not sure if CNO is the only one. I'm sure CNO is the only one they tested, but I'm not sure if it's applicable to humans. But the fact that they kept mentioning like, oh, like in the future we want to test, like we don't know if this is applicable to humans, maybe like not. Yeah. Sarah,
Can you go to the slide on the Markov model and describe the picture?
this one.
Yeah, that one.
Okay. So just like explain the actual picture. As you can see in the normal Markov model, the state is like clearly observable and transitions happen directly between states. Versus like the hidden Markov model, it looks a lot more confusing. Basically, you just can't observe each state directly. Each hidden state can generate multiple possible measurements. As you can see, like state 1 can lead to A, B, or C. And measurements are like just probabilistic, so like the same state can preserve many different observations, but you can infer the hidden state indirectly from patterns in the observations. And those are hidden Markov models are very important in a lot of neuroscience experiments and in this one. Yeah.
All right, that's the attendance form if you need to fill that out. Thank you. Also, make sure to push in your chairs and clean anything up.
Sarah's up.
Next week is Sarah's presentation.